Thanks again for attending this first Vesiculab webinar today. It's a pleasure to have our guest speakers, Oscar Wiklander, as well as André Görgens, both working at the Karolinska Institute, and to basically discuss their work on the engineering extracellular vesicles, how they can be applied to different applications, including therapeutics, as well as analytical purposes. And before that, I will give a small presentation about Vesiculab and their activities. So let's get on to it. Yeah, very briefly about Vesiculab. It's a company that started only a year ago, and with the goal to bring some innovative solutions to assist the extracellular vesicle workflow. And these include dyes to specifically label extracellular vesicles, as well as reference materials for EV analysis. These will be discussed by Oscar and André later. A sample buffer for extracellular vesicles preservation, that will be discussed by André as well. And finally, size exclusion chromatography spin columns for the preparation of extracellular vesicle samples before analytics or functional assay. And this aspect I will briefly discuss here. So Vesi-Sec micro or size exclusion chromatography column that are assisting the sample preparation to remove unbound dye molecules, as well as antibody that is present after the staining of EVs before flow cytometry, fluorescent NTA, fluorescent MRPS, as well as functional assays like imaging. So all these unbound material are creating background noise that reduces the sensitivity of the measurement. And one way to increase that sensitivity is to quickly remove these molecules, unbound molecules. So over the past few months, these columns have been extensively used, and their efficiency has been demonstrated. Here, Christian Plosser at Marburg University used the column to remove unbound antibody, anti-CD63 antibody used to label EVs before nano-flow cytometry, as well as a lipophilic membrane dye. And in both occasions, it was able to pick up many more events by reducing the fluorescence background, the noise in the signal. A bit later in the year, Rachel Butler, who's a PhD student at Aston University, compared the efficiency of the spin column approach compared to the gravity flow column approach, and she was able to get equivalent result to gravity flow size exclusion chromatography. But instead of spending half an hour doing so, she could get it in 1 minute spin. And more recently, there was this study from Anna Jackson at the University of Nottingham, where she basically used Vesi-Sec micro to remove unbound dye before imaging, and contrasted with the ultracentrifugation approach that she usually applies, and demonstrated that she could get better results faster by removing Antibody using this approach. So in short, we are at Vesiculab, we are building a portfolio of innovative solutions, and now I leave it to Oscar and André to present the engineered EVs that they have developed and are now part of product range as well. So I give the floor to you. André and Oscar, can you? Thank you.
Yes, thank you so much, Dimitri. Thanks for the invitation and the opportunity to share some data. I hope it will be a nice discussion at the end as well, because it's an ongoing thing to do research in this area, of course. So yeah, today we're going to talk about both EVs specially engineered for targeted drug delivery and also some research applications. So I'm going to start briefly before Oscar takes over. Quick conflict of interest disclosure. And yeah, the main topic of our lab is bioengineering of EVs for therapeutic applications. And of course, that's kind of like a simplified process, but developing such a process for especially different therapeutic applications requires a lot of, let's say, decisions and also a good quality decisions. So of course, there's lots of choices to make, like which cells to use, how to culture the cells, how to purify or how to isolate the EVs at the end, how to store them, and what assays to use at the end to dose or characterize the EVs. And of course, what has been shown over the years while we did all these projects about therapeutic applications and using engineered EVs, it has been incredibly useful to have good analytics in place that can tell about these decisions in the process and also the final EV batches to make sure what we use for experiments and mouse studies are really having a certain quality or meeting a certain quality threshold. When we talk about EV engineering, there are basically two ways to do that, either by mixing of molecules or drugs with EVs after they have been produced, or what we are doing in the project that we are talking today and mostly and also in most other projects is endogenous loading of EVs. And in a nutshell, it's expressing an engineering construct consisting out of an EV sorting domain and a protein of interest in your cells, and these genetically manipulated cells then release EVs that are made for purpose for a specific therapeutic property. For instance, introducing a targeting protein or on the outside, or loading an RNA in the inside, or combining of different approaches in that sense. And the first time this, or one of the first things that have been tried in terms of endogenous loading or engineering was basically CD63 EGFP tagging of EVs, or fluorescent protein tagging of EVs with the CD63 fusion protein. And this basically leads, since CD63 is going at high abundance to EVs, to the production of EVs that intravesicular have a fluorescent tag. And of course, that's very helpful because especially if you're trying or setting up a new method, it's very helpful that you know that the EVs, the things you're looking for, are actually fluorescently tagged, or whatever is fluorescently tagged is an EV. So we produced in this study early on from THP1 cells that are having a CD63 EGFP fusion protein fluorescently tagged EVs, and then used that for different methods, establishment or optimization, but also for imaging flow cytometry establishment, which is a specific type of flow cytometer that is, due to different reasons, quite sensitive compared to like conventional flow cytometers. And we early on succeeded then to really identify scatter low fluorescent positive EVs in this with this technology, and it was incredibly helpful to have these CD63 EGFP engineered EVs as reference material throughout. And just one experiment I want to show to underline the usefulness of having a fluorescent tag. So these THP1, CD63 EGFP cells were completely taken from cell culture, so cells and the complete supernatant without any modification, and run in the imaging flow cytometer. And you see, if you look at the scatter versus fluorescent plots, you see there are different fluorescent populations. And if you then have an imaging flow cytometer that we use here, you can, of course, validate on the images to some extent what these populations refer to. And you see this top one, R1, is the cells. And if you go down the scatter axis, you actually see that the R2 gate is probably debris or apoptotic bodies. And if you go down and zoom in here to the lower populations, way below a cell's scatter equivalent, you see there are different populations. Then at the end, this R5 gate, you can't actually pick up any bright field or scatter signals, but single fluorescent dots. And that was then again evaluated to some more extent when we went through an EV isolation procedure, in this case, a differential centrifugation scheme, and then actually measured the respective samples after 2,000 or 10,000 G centrifugation. Again, and you see it cleans up more and more in terms of losing the scatter high events. But then if you do an 0.2 micrometer filtration step, you see that what you are left with at the end is this scatter low fluorescence positive population here marked as R5. And that's also the population you mainly recover with after ultracentrifugation. So this shows nicely that it's helpful to have fluorescently tagged EVs or cell lines that produce those EVs as reference material to actually follow the EVs through a process to see what happens in each process, to quantify at what step do I lose how much. Then you can, of course, modify the process accordingly and optimize it over time. And this is what we did kind of all the time when we set up a new method. Then further on, we also used this model of using fluorescent EVs, either unstained or control antibody stained and stained with antibodies, and actually also used this model of fluorescent EVs tagged, stained with fluorescent antibodies to optimize the EV labeling procedures a bit more. Then later on, we used the Amnis cell stream instrument, which is similar to the image stream, but has some differences in terms of how it processes the data. Anyways, this was again a new instrument for us, so we again used our fluorescently tagged EVs. Meanwhile, we had moved on to neon green tagged EVs instead of EGFP because it's brighter. And then also qualified this and used it in different ways to here, for instance, to stain with tetraspanin antibodies against neon green tagged EVs to estimate the engineering efficiency in our different preps. And then, of course, we did our homework again, did different controlled serial dilutions, titrated our antibodies, looked into assay variability, and for some antibodies also validated them on knockout derived EVs. So having talked about these EVs as fluorescent, as reference material and showing some examples how they can be useful. Of course, I mentioned that we are working on developing EVs for therapeutic applications. So I'm going to hand over now to Oscar, who will talk a bit more about an actual really cool project that started a long time ago. And I'm really happy that this is now finally out in the published, and yeah, looking forward to hear your story.
Thank you very much, André, and thank you, Dimitri, a lot for inviting us to discuss a bit on our recent paper. As André mentioned, it's taken us quite some time. It's been an ongoing project for almost 10 years that we're very excited that we now can share it with all you and the people wanting to read the paper. So we're discussing then antibody-displaying EVs for targeted therapy. We call them Fc-EVs because they bind the Fc part of antibodies. I would like to just take a few steps back, how we came up with the idea. So there's plenty of evidence of using engineered EVs for targeting. Back in 2011, Lydia Alvarez published a very nice paper in Nature Biotechnology showing brain targeting by introducing then a fusion construct with the LAMP2B RVG, this rabbi viral glycoprotein, and we're able to show brain targeting. Since then, similar systems with fusion constructs with one EV sorting domain and then having this used to a targeting moiety, targeting various tissues in the body have been shown and published. So it's not new that EVs can be targeted and go to the site of action that you want them to go. But here at the same time, what we wanted to do was to see, can we use something else? At the time I was, so I'm an oncologist, and this was during my oncology training. So reading up on the very cool field of antibodies that are used for therapeutic applications in oncology. And there are several different targets, such as HER2, VEGF, EGFR, PD-L1 and PD-1 now coming very heavily to be used in oncology as well. So these antibodies then, of course, have a therapeutic effect. But what we were thinking then was they also, of course, are highly optimized for targeting to go to the antigen where they're going to have the therapeutic effect. So we were thinking, could we then combine the effect of a therapeutic antibody, still have the therapeutic effect, but also use its targeting capacities and display this on EVs to get the EVs to go to the site of action that we want them to go to and deliver a cargo. So that's what we set out to do. And the main idea here is using a similar concept then, as has been published and we and others have done before, using a fusion construct. And we did an optimization. We went through and screened different EV sorting domains and different Fc-binding domains to get the antibodies to be displayed on the surface. And after the optimization, we ended up with a leading construct. In our case, it was CD63 combined with the Z domain, which is derived from protein A, which is from Staphylococcus aureus bacteria. So first thing that we wanted to do was, of course, to characterize this. Do they work? Do they change? So we first, if you look here at up to the left, graph A there, it's just an NTA showing that the size and size distribution and particle count of the EVs did not seem to be affected, whether we had this Fc-binding construct on the EVs or not, or if we had an antibody there or not. And then, of course, most importantly would be that the antibodies are actually displayed on the surface. So as you can see here in B, it's we had a reporter, fluorescent reporter, as Antje mentioned. We started using M-neon green, and we used this for these Fc-EVs as well as the control EVs. And then we combined them with the APC positive antibody. We incubated the Fc-EVs together with the antibody and then did a size exclusion chromatography. And as you can see on B there in the upper graph, we can see that they co-elute to the same fractions as the expected EV fractions, both the antibody and the EVs. And in the lower graph there, you can see that the control EVs elute by themselves at those fractions, whereas the antibody then, when there's no Fc binding part, elute at much later fractions. We also did characterization by imaging flow cytometry, as André introduced to you just before this, and I think he will talk a bit about that later on as well. And we also did a lot of other characterization steps. So next, we wanted to see, of course, can this then be used for targeting? Can we use the system to deliver the EVs to cancer cells? And we had two proof of concept models, one being Here to the left, HER2 positive breast cancer, and then PD-L1 positive melanoma. So in HER2, in the clinics, we use trastuzumab. It's been an antibody, a HER2 targeting antibody that's been around for 20 so years, highly efficient, binds HER2 very well. And we put this then on top of the Fc-EVs and saw that, and then put the Fc-EVs with the trastuzumab antibody on top of HER2 positive breast cancer cells, and saw that they then had an increased uptake of the EVs, about 300 to 400-fold increase. This did not happen when we just had the EVs without the Fc-EVs without an antibody, or with the control antibody. And of course, the control EVs without having the antibody displaying properties did not show any increased uptake either. Similarly, PD-L1, so PD-L1 and PD-1 have sort of revolutionized cancer care the last 5-10 years, acting as immune checkpoint inhibitors, as most of you probably have heard of. So they block the inactivation. So PD-L1 and PD-1 act to suppress the immune recognition of cancer cells, and the antibodies block the signal, and thus reactivates the immune system to recognize tumor cells. So here we used the PD-L1 antibody. We tried a few different ones and saw that the best effect we got with the PD-L1 antibody called atezolizumab, also clinically used. And we tested this on melanoma cells. So when the Fc-EVs displayed atezolizumab, we had about a 500-fold increased uptake as compared to the same Fc-EVs without antibody. So this seemed to be a clear targeting effect in vitro. Going in vivo, we used the same models. Here I'm showing for the melanoma setup. So we had mice bearing the same malignant melanoma. And when we had a palpable tumor, we injected these with a single injection with Fc-EVs, either with a control antibody or displaying the PD-L1 antibody, this atezolizumab, or without any antibody at all. And what we could see then was that we had a clear increased uptake also in tumor tissue after systemic injection. So we had about a 50-fold increased uptake. And what we were very happy and pleased to see was that this uptake seemed to be sustained over time. So following a single injection, up to 72 hours post-injection, we still had a significant difference between Fc-EVs with the PD-L1 antibody as compared to just having a IgG control antibody or just Fc-EVs by themselves. So this seemed extremely promising. So then we wanted to choose a cargo to deliver with this system. And a few years ago, we published an optimization of loading doxorubicin into EVs. So that made us also think that doxorubicin could be the chemotherapy, the cancer drug of choice. It has some good properties. One is that it's autofluorescent, so it's very easy for us also to actually see how much we load. And we had already, in this publication, tested it on melanoma cells and saw that when doxorubicin is encapsulated into EVs, it becomes much more potent than giving naked doxorubicin. So here, giving the same dose of doxorubicin, we saw that EVs encapsulating doxorubicin were about 200-fold more potent in killing off malignant melanoma tumor cells as compared to just giving it by naked doxorubicin. So going back to the reasons and why we want to do this, because I think that's important to keep in mind at all times. Of course, this is proof of concept models we're dealing with here also. And targeting to start off with, the main idea of any targeted therapy is, of course, to maximize the effect and hit the bullseye of the tumor, while at the same time minimizing off-target side effects. Here, we put yet another layer on this by thinking that we can also, through this targeting, have a synergistic effect. So if we use this PD-L1 antibody, as I mentioned, it's a checkpoint inhibitor, we will have an effect of the antibody. And this will also help us get the EVs to target to the tumor site. And if we then can load the EVs with doxorubicin, this should have a cancer-killing effect also at the tumor site, which will then increase the release of neoantigens, would further activate the function and enhance the function of the checkpoint inhibition by the PD-L1 antibody. So then we, of course, set out to try to use this as a therapy. We loaded Fc-EVs with doxorubicin and had them displaying the PD-L1 antibody. And of course, plenty of different controls for this setup as well. But as you can see, what we found was that this therapeutic combination with Fc-EVs with PD-L1 antibody and doxorubicin had a clear therapeutic benefit with decreased progression of the tumor over time and a clear survival benefit. So we had 100% of the mice surviving in the Fc-EV PD-L1 antibody doxorubicin group, as compared to the PBS only control group, where we only had 30% of the mice surviving. So we then also set out to see, okay, how long can we have this effect for? So we did another setup where we tried to increase the time of this highly aggressive tumor model. And here as before, we started injecting the mice with systemic injections after we had a palpable tumor, and then injected every third day. And as you can see, also for a longer time period, we had a clear benefit with a reduced progression of the tumor size. And here up to day 35, we still had an 80% survival rate in the therapy group, whereas the control mice just receiving PBS had all died off at day 20. Fc-EVs with just a control antibody, but still loaded with doxorubicin, had some effect, but it was not at all as close to when we also had the PD-L1 antibody. So here they survived until day 26. So then, what can we use this for? For me, it's an extremely interesting platform, even though it's been 10 years for us to get this out and published. It's I still consider it sort of a generation 1. I think it can still be further optimized and improved, of course, either by us or you listening or in collaboration. And I hope it will be available as a therapeutic platform in the future. So of course you can pick the antibody of choice. You can get these EVs delivered to pretty much anywhere in the body that has an epitope that we have or an antigen that we can target with an antibody. You could potentially use more than just one antibody. We know here that we have about 100, so the average was 105 antibodies per EV when we analyze that. So you could have multiple antibodies. You could use bispecific antibodies as well. It's also the last few years, the big thing in oncology now is ADCs, so antibody drug conjugates, which you then could use as well to deliver multiple cargos. And it's not just limited to an antibody. You could use any kind of Fc fusion protein as well, as long as it has an Fc part that can bind to the Fc binder. At the same time, it's also very versatile with the choice of payload, and that's just limited by one's creativity, I think. Here we've been looking into chemotherapy. There are, of course, multiple different cancer drugs. Sometimes they're too toxic to use, but here we might be able to get them targeted in a better way and have them effective and with much less toxicity. You can also consider, of course, gene therapy molecules such as siRNA or mRNA. And it can, as I mentioned, be probably further developed because this, as it is now, it's in a way working as an ADC, an antibody-guided delivery of a cargo, even though this is more versatile. But you can also consider EVs having an intrinsic effect. There's multiple evidence of that as well. For instance, when it comes to MSC, EVs has been shown in many settings to have immune modulatory effects. We know that NK-derived EVs or mature DC-derived EVs can seem to have a natural cancer-killing effect. So we could consider having, depending on our target and our therapeutic application, we could also consider using and choosing the right source for the EVs as well. And in addition to all the therapeutic applications, we can, of course, also consider using this as a very interesting reference material. And I'll hand it back to you, André, after that.
Yeah, thanks a lot, Oscar. So I'll walk you through some more details of that aspect. As you already saw in brief in Oscar's presentation, we used imaging flow cytometry with both the CD63 amnion green control EVs and the CD63 amnion green Fc-binding EVs. And basically, just wanted to emphasize that it was, of course, incredibly helpful for this project to have a method and material to quantify at what or to titrate how much antibody we could use to load EVs to saturation and to quality check batches or EV preparations before we would use them for experiments. So that's generally a very nice use case of how fluorescent EVs or functionalized fluorescent EVs, such as these Fc-binding EVs, could be used. And of course, further on, as a potential reference material, Fc-binding EVs, of course, leave a lot of room for ideas such as compensation controls, positive controls for different assays, or general material to study antibody binding to EVs and their stability and their impact on cellular uptake and so on. So that's a quite versatile material, I would say. Just wanted to emphasize one more aspect of fluorescent EVs as reference material, which is EV stability. And of course, for this project, but also for many other projects that we did in the last few years, it has been incredibly important that we have reproduced EVs in large amounts for mouse models, for instance, and then make sure that these EVs are stable between the point of production or when they are measured to the actual injection into mice or use in cell-based assays. And pretty early on, also like 8 years ago, I think, we started doing stability studies, and also here again used fluorescently tagged EVs, CD63 mNG, together with other EVs, and basically stored them in different buffers, different temperatures, different conditions, and ran all assays we could do for up to 2 years. I just want to cut this short to emphasize that stability is quite important. We saw quite a drastic loss of EVs stored in PBS as a control compared to some of the buffers, and the best buffer we could identify in this study was we called PBS hat. Basically PBS with HEPAS, albumin, and trehalose added, and minus 80 degrees was showing the best results. Again, within this study, at the end, we also used imaging flow cytometry and fluorescent EVs as reference. And one simple experiment showing the power of this and the importance of stability or stabilizing buffers is this experiment where we use fluorescent EVs, diluted them in different buffers, and directly measured them by imaging flow cytometry, counting the concentration of fluorescent amnion green positive events. And you can see that if you do that in PBS hat, you get pretty nice and stable values, while if you do that in PBS, you get roughly 70% less events recovered. in your instrument, and you even have through subsequent wells a further loss of EVs. To cut this story short again a bit, the major loss that we saw here, we think will be attributed to plastic adsorption or surface adsorption during the handling of EVs. So we have no evidence that the EVs are actually destroyed, but they are nicely retained if you have the head buffer compared to other buffers. I'm adding this because I'm getting asked this a lot all the time when I show this. So we also tried all the low bind and differently coated tubes that we could find. And all of those led in PBS to drastic losses of EVs over, in this case, 7 days. While in the head buffer, we don't see any of that really, at least not significantly. So it's not the tube, it's more the buffer in this case. At least we didn't find a tube where this doesn't happen. And EVs in that buffer stored at -80 and going through up to 5 freeze-thaw cycles are also, in terms of concentration of fluorescent EVs in those samples, pretty consistent over time. So that enables, of course, a lot of nicer and more high-quality experiments using this buffer. And after actually we published this paper, we got a lot of incredibly positive and feedback, and also a lot of questions also around how to prepare this buffer and how to use it. So I'm glad that we teamed up with Dimitri to commercialize it as well. So it's available as Vesi-safe, and of course you can create it on your own, but if you want to take a shortcut, you can order it and get a 10x solution that is equivalent to the PBS head solution we used in that paper. And if you want to try this on your own, you can basically take EVs as I showed and dilute them in PBS versus this buffer, 2 different dilutions, and you see if you compare PBS versus Vesi-safe or PBS head, at 2 different concentrations, you lose roughly 90% in these experiments. So that's something you can easily verify within an hour or so in the lab if you want. Mentioning CD63 neon green and CD63 Fc neon green EVs as reference material, it's of course important that those are available to the community and not only produced in our lab without the knowledge and the material shared. So I'm also happy that Dimitri was interested in commercializing those. So they are available now as Vesi-Ref CD63-mNG and Vesi-Ref Vesi-Bind Fc-mNG. It's the same control and Fc-binding EVs we used throughout the study that Oscar showed. Here's some more data on our cell stream where you see that if you add, and this is the same that we added here, it's an isotype control human IgG1 antibody that nicely binds to the Vesi-Bind Fc-EVs, but not to the control Vesi-Ref CD63-mNG EVs. And if you just use the CD63 anti-human antibody, you get pretty much the same pattern for both. Then, of course, reference materials need to be available to the community, but they also need to be, and they need to be stable, of course, and fulfill some criteria that make them useful, but they also need to be well characterized. And that's where you and others come in, of course. And Dimitri just shared with me actually yesterday and a few days ago some preliminary data that we received after sending these, in this case only Vesi-Ref CD63-mNG EVs to different companies that offer or that produce analytical instruments. Here's some data from the Spectrodyne ARC. Showing some quantification of mNG positive versus mNG negative EVs in this sample, and also staining with CD63 antibodies. Then here's some data from the Leprechaun from Unchained Labs. That's an image. I'm not 100% familiar with all the details, but just wanted to give you some impression here, some brief examples how it can look in different applications. So it's an imaging-based technique, and they can actually quantify CD63 positive versus CD63 positive labeled, antibody-labeled mNG positive populations, and they come up with an engineered EV percentage of 75%, which roughly matches the data we generated on the same batch. And here's some data from Beckmann-Coulter measured on the Cytoflex Nano, also showing quite interesting first impressions of what can be done also with diameter assessments, size calibration. And yeah, I guess this is just a first impression of what we could do with this. We are more than happy to, of course, share those EVs or collaborate with others to get more data on this, because if we get more information and more characterization done on a specific reference material and also benchmark it or compare it to other reference materials that are available, we will have more value from this for everybody. So yeah, to quickly summarize, we showed you that Fc-EVs are really cool to target and deliver drugs to tumors, and are probably a versatile tool to use in many other contexts, as Oskar showed. I showed you that to set up an EV production process, it's incredibly useful to have reference EVs and good analytics in general. I showed you some examples how to use fluorescent EVs and Fc-binding EVs, and I mentioned that we are commercializing those with Dimitri. And yeah, I'm thankful to everybody who contributed, of course. Feel free to contact us if you want to discuss more, collaborate, whatsoever. And yeah, looking forward to a good discussion. Thanks.
Thank you very much, Oskar and André, for this very, very insightful presentation. We have quite a few questions on the Q&A and the chat, so maybe I'll start with the two on the Q&A. Started with Dan, who was asking, what quality controls do you have in place for the loading of internal EV molecules?
For loading internal EV molecules.
Like exogenous loading, I guess.
For the exogenous loading then, yeah, because of course here we use both systems, both exogenous and endogenous. I assume then meaning to the electroporation when we're loading the payload then.
Yes,
yeah,
it was in presentation. Yeah.
Yeah, so that's a good question. So what we do then is since here doxorubicin is autofluorescent. So the loading here is based on reading the fluorescence of doxorubicin. So it's quite simple to control for that, and we load it, and then what we do after loading is always to do yet another size exclusion chromatography to get rid of anything that's unbound or not loaded into the EVs. Worth noting is, of course, also that it's not 100% stable, which we also show in the paper, that over time you do get some of the payload going out of the pores we created through electroporation. This is probably not only a bad thing. So we know that the half-life is extremely short for the EVs, and we know that we can see tumor that they start going into the tumor after systemic injection within a few minutes. So having it released is probably a good thing then, so that we get it into the environment, the tumor environment, and also getting a bystander effect, which has been shown when it comes to the ADC field, for instance, that we have extremely good effect by getting the bystander effect as well. Yeah, that was a long answer to that question. Sorry about that. I'll try to keep it shorter next time.
No worries. Thanks, Oskar. There's another one actually related. It was the loading efficiency of doxorubicin and of monoclonal antibodies on the Fc-EVs.
Yeah, as I mentioned, we did quite a lot of tried in different ways to characterize how many, if we start with the antibodies per EV. So we ended up showing that roughly between 100 to 200, I think that we state 105, because that was based on a more recent analysis using what is called now this single I forgot
Single
that.
particle profile.
Single particle profiler, yeah. So it's a new technique also published earlier or about a year ago now, I think, from also a group at Karolinska. So it's basically using confocal microscopy and detecting small molecular changes over time. So they're able to actually go in depth and look at single molecules if they're fluorescently tagged then on the surface of EVs. So using this system, we were able to detect, we know there's a span. Some Fc-EVs will probably bind fewer and some might bind more. As it's not a synthetic product, there's going to be changes, but and differences. But in average, it's about 105. And when it comes to loading efficiency of the exogenous loading into doxorubicin, we based most of the data from our previous publication there in 2021, published on Christmas Eve that year. So it's fairly low amounts, I would say, that we load. So it's always a cutoff. You can load a lot, but then you lose the function of the EVs. If you tune up the settings for the electroporation, a lot of the EVs will just get destroyed. So there's a clear relation between loading efficiency, and you have to find a good balance where you have what you think then is good enough loading, and also have, because you want the EVs to be functional, of course. Yeah, but all the numbers are in the paper, of course. Yeah.
Thank you. I'll just go to the chat. We have quite a few questions. So again, I'll go back to the very beginning. I had a question from Abdul saying that very early on in the presentation, you were talking about delaying experiments. Does it reduce the yield and quality of EVs? Or I think they were going back to the stability.
Yeah, I'm not 100% sure how it's meant, but I guess just imagining you would produce all EVs fresh and use immediately, or how we did it at the very beginning, that's lots of work, lots of late night shifts, and not feasible in the long term. So finding a buffer that stabilizes EV batches so you can produce a few big batches and then use another day has been a game changer for sure.
Yeah, definitely. And for using it as a reference material or as a therapeutic tool, you will need to have the ability to store it. Before we always plan the production and the animal experiments very tight, and that can be done when you're working on small scales, just for proof of concept studies. But if you want to do something and produce a reference material or a therapy, that needs to be, if you can store it, that's an extreme upside. We know that from different like CAR-T cell therapies or whatever, that it's otherwise, if you have to start production right away, that could take months, and the patient might be dead then, or whatever you want to analyze might not be there for that, when you could just take it out of the freezer to use it as a reference material, for instance. So storage is very much a key. I agree with André.
Then we have a question about what is the added value of Fc-EVs compared to antibody drug conjugates?
Yeah, no, I think that's a great question. So when I started working, or when I had the idea, I wasn't that aware of the ADCs, and so now it was just ending yesterday. Was this ESMO conference, the big conference in Europe for all oncologists, and the big buzz is ADCs. It was at ASCO, the American Society of Oncology, as well. Earlier on this year. So it's ADCs is what's big right now. And if you just give it with a single antibody and a single payload, then I don't really see much, or we don't know, but one benefit would be, of course, you're using a system. So EVs have been developed through evolution for us to have ability to shuttle different kind of molecular clues between tissues. So I think it's something that has been developed that we're utilizing. But I think that the main idea is that it's much more versatile, that you can choose your payload. You can have different antibodies. And as I mentioned in my presentation there also, if you can get yet another layer by using the intrinsic effect of an EV, depending on the cell source, then I think you can have a much more multi-modular therapy than ADCs, which are restricted to only having one antibody and one payload, pretty much.
Reasons are needed, obviously. What about biocompatibility? Any data on that or opinions, findings?
I think it's an extremely important consideration. So first of all, we need to just consider that patients are getting blood transfusion on a daily basis all the time, all over the world. And there are, of course, getting EVs then too from another person. And those are usually safe. HEC EVs, as we're using here, as cell source, are used also as a cellular therapy and have been developed or have been okayed to use in clinical trials. So pretty much any kind of cell therapy, if you think that the cells are safe, I would assume that the EVs are, but of course they would have to go through all the regulatory testing before actually start treating patients. That is extremely important. That has to go through phase 1 clinical trials and so on. But what we've seen so far, so in our setup, we were giving then a human-derived EVs, HEC EVs, to mice, giving them repetitive injections. We did not see any signs of toxicity. It can be hard to evaluate in a mouse that has a tumor and, of course, have symptoms from its tumor, but we did not see any difference between EVs here for the mice. And what other people have shown is that it seemed to be safe. It's also with the early clinical trials, 2004, 2005, with DC-derived exosomes that were tested in melanoma and non-small cell lung cancer, were also shown that they were safe in humans. They were not very effective at the time. They were also using immature DCs as a source, which with our current knowledge might have been better to have used mature EVs, mature DC EVs. But so they seem to be safe, but of course this needs to be tested in more clinical trials.
And I think you answered the following question about your cell of choices, or X cells. So we're going to move on to the next one. How efficient is the antibody-charged EVs at delivering the payload into the target cell? The efficiency of delivering the payload, so really the targeting of it.
Yeah, so we did not really look at exactly how much of the payload goes to the tumor. So one could consider that one way would be, so we looked at how much of the EVs go there, but it might be also an interesting experiment to see what % of what you inject goes to the tissue of the payload actually, and not just of the EVs, because we did all the biodistribution studies were done with unloaded EVs, and then we saw that it was a huge increase. It's important to consider also that targeting doesn't mean that we get 100% of the EVs going there. That's clear. So we have it's a few percent as compared to less than 1%. So it's a clear improvement, as I showed. It's about a 50-fold improvement in targeting efficiency, but there's still a lot going to liver and spleen. If if we can get the payload to go 50% more, or 50-fold more, in my world, that's still extremely important because we can still increase the dose dramatically. So as a reference, doxorubicin is not even used in the clinics to treat melanoma because you would need the dose to be so high, so it would be cardiotoxic before you could ever use it. But in this setting, we can increase the targeting so much so it actually becomes efficient. Also in melanoma, which is known to be chemotherapy insensitive otherwise.
Sounds good. We had a question about scale. Can EVs be manufactured at scale when it comes to obviously clinical testing?
Yeah, I guess the answer is yes. It's not a secret anymore, and there's different ways to scale up quite nicely with bioreactors, and also there are industrial size tangential flow filtration or size exclusion columns available nowadays. It's just a matter of costs, basically.
Yeah, and I think it's as you say nowadays, because it's also that there's been a lot of developments by a lot of different groups on not only so production is one thing, right? You can just have a big tank and produce a lot of EVs, but then it's also it used to be that the isolation process could be quite a bottleneck of the production of EVs. Back in the day, ultracentrifugation was the gold standard. Might be sometimes too now, but that would probably not be feasible in a clinical setting to do ultracentrifugation with everything. But with all the new filtration isolation systems, I think that is also pretty rather easy to scale up. So it's definitely scalable. Still costly, but everything's costly at the beginning.
Yes, probably taking a leaf from gene therapy anyway, the similar pathways.
Yeah.
Maybe just to finish, just conscious of the time, we won't have the time to go through all questions, but it's one that is a bit technical. Maybe can the antibody be added to the conditioned media before SEC isolation?
Yes.
Yes.
Yes, it could. We're now when we're doing other experiments with it. That's what we're trying, and we're comparing it. It shouldn't be an issue to do that, because we previously, just to make sure that we had everything completely controlled, we had them first isolated, and then we did, as I mentioned, yet another size exclusion to get rid of antibodies that were unbound, or we made sure to use lower amounts of antibody not to saturate the binding sites, the Fc binding sites on the EVs, but they can definitely be used at the same time.
Sounds good. So I'm very sorry for the many questions that were asked and that we won't have time to address during the webinar. But I will send them on to Oscar and André, and we'll try to get back to you. Thank you very much again to you, André, Oscar. It was a pleasure to have you and to hear about your research. And thank you to all the attendees for taking the time, and feel free to contact us if you have any questions. Thank you very much.
Thank you.
Yes, thank you very much.
Take care. Bye-bye.